Happy World Carnivorous Plants Day! My name is Emmi Kurosawa. I'm the current Vice President of the New England Carnivorous Plant Society in the US. Today, I would like to share with you my recent expedition to Australia. The main purpose of this expedition was to see the Utricularia dichotoma complex in person. Professor Jobson and Baleeiro recently split Utricularia dichotoma complex into 8 different subspecies by DNA analysis. Usually people see a distinct morphological difference and then try to look for DNA sequences that were responsible for it. However, this case was the opposite. The DNA information said they were different, but morphologically the differences were not as black and white. So I had this wonderful opportunity to travel with the world-renowned botanist, Dr. Rob Gibson. Dr. Gibson and I visited the known Utricularia dichotoma sites in New South Wales, Australia, to see if we could find the morphological differences that were described in the paper, and also if we could actually identify some populations that were not genetically classified yet. I hope you enjoy. Before I dive into the story of our adventure, I would like to briefly introduce the genus Utricularia, the suckers. The genus Utricularia belongs to Lentibulariaceae with 2 other sister clades, Pinguicula and Genlesia. There are more than 250 species worldwide, and they are separated into 3 genera and further into 23 sections. The genus Utricularia, in my opinion, is the most fascinating genera of all carnivorous plants because it uses its unique suction strategy to trap small aquatic zooplankton. And this suction speed is the fastest movement in the plant kingdom. The snap trap of a Venus flytrap takes about 100 milliseconds to close, whereas Utricularia traps open and close in just 1.5 milliseconds, almost 70 times faster than the Venus flytraps. They are cosmopolitan and are found on every continent except Antarctica. Also, Utricularia giba has one of the smallest genome size next to the sister clade Genlesia. This is mainly achieved by having only 2% junk genes as opposed to 98% in humans. Because of these unusual properties, numerous scientific discoveries have been made on Utricularia. I am only showing here a fraction of all the amazing facts of this genus. Here is something about evolution. According to the ICPS website, carnivorous plants appeared first in the form of nepenthes about 85 million years ago. Pitfall traps like nepenthes, Darlingtonia, Cephalotus, Sarracenia, Heliamphora, Papalanthus, Catopsis, and Brochinia, and also flypaper traps like Drosophilum, Drosera, Biblis, Roligula, Philcoxia, and Triphyllophyllum. They all occurred multiple times in the plant history. However, the suction trap strategy of Utricularia so far only occurred once about 31 million years ago. Let me explain here the suction mechanism of Utricularia traps. So think of the trap as big sack with a door. These bladders constantly pump water out of the traps, creating a suction within. The door has several trigger hairs. When the zooplankton bumps into these hairs, the door contorts. Now the suction is broken. What happens next is that the water surrounding the bladder gushes into the trap with the zooplankton. As the pressure within the trap equilibrates with the surrounding pressure, the door shuts tight behind the poor prey and the digestion begins. Lastly, I would like to show different forms of Utricularia. Utricularia is flamboyant and takes many different forms and occupy almost all possible niches: terrestrial, aquatic, epiphytic, lithophytic, and rheophytic. Epiphytic meaning these plants obtain water and nutrients from the air, and lithophytic meaning they grow on rocks, and rheophytic meaning they grow in fast-moving water. Just to show one example for each, for the terrestrial species, we have here Utricularia resupinata, which ranges from Canada down to Central America. For one aquatic species, we have here Utricularia inflata, which is native to the United States, one of my favorite Utricularia. For epiphytic species, we have this beautiful Utricularia campbelliana. Epiphytic species generally have larger orchid-like flowers and it's very showy. For lithophytic species, we have here Nephrophila. Thank you, Fernando, for the photo. Lastly, for rheophytic species, we have here Utricularia neotioides. Here, we have the protagonist of my talk, Utricularia dichotoma. Utricularia dichotoma complex belongs to the section Pliocasia under the subgenus Polypomphalix. They are terrestrial herbaceous perennial plants that grow in Australia, New Zealand, and New Caledonia. Here we have the whole Utricularia dichotoma complex. The plants on the left side are considered species. We have Ameliae, Speciosa, Fenchamiae, Barkeri, Grampiana, Oppositiflora, Buguliholiae. Buguliholiae has two different subspecies, subspecies Buguliholiae and Orientalis. The plants on the right are the Utricularia dichotoma subspecies, which were recently split into 8 subspecies. Subspecies dichotoma, Monanthus, Novae Zelandiae, Aquiliona, Fontana, Novae Angliae, Oxaliensis, and Maritima. The plant names shown in red are the ones we think we were able to recognize during this expedition. Here is the overview of the expedition. Dr. Rob Gibson and I visited 8 sites spanning about 800 kilometers along the coastal region of New South Wales, from Sydney to Evans Head, looking for Utricularia dichotoma. Then I said goodbye to Dr. Gibson and moved on to my next destination, Tasmania. In Tasmania, I met up with my friend Lisa Robbie, who drove me all around to see 2 additional species. Before I start, please let me explain first how to morphologically distinguish dichotoma complex. These 4 key features are the most important clues to determining Utricularia dichotoma complex: bracts and bracteoles, leaf shapes, spur shapes, and corolla colors. Bracts are the structures that are attached to the base of the peduncle, and bracteoles are attached to the base of the pedicels. So pedicels would be holding the flowers, and then the peduncle would be holding the whole flowers and the pedicels. Depending on if they are attached at the bottom or in the middle to the pedicels and peduncles, they can be sometimes the key to determining the species. Also, the existence of a spur at the bottom of the bracts and bracteoles, so these little protrusions, and if they are gibbous, meaning little swollen or not, is also an important key. But all these differences can be very variable between individuals as well as between populations. The leaf shapes are highly affected by the environment in my limited experience, but some species stay relatively the same and can be used as taxonomy keys. In the paper, the leaf shapes were described as either linear, lanceolate, ovate, or ovate. So here in this case, this would be linear, I would say, and this would be somewhat ovate. And the spur shapes and length, like these, can be also a taxonomy key. The word spur is used for describing two different tissues in the paper. So the spur here is the little nectar-holding organ attached to the corolla. Spurs, I described above, are the little protrusions, sometimes seen at the bottom part of the bracts and bracteoles. So let's not get these two confused. Corolla is the flower. Utricularia has two lobes, upper lobe and the lower lobe. They are also called lips here. So in Utricularia dichotoma complex, upper lip is tiny compared to the big and showy lower lip. They are highly variable and generally not a good way to determine species, but sometimes specific features such as the existence of freckles or dark veins on upper corolla lip can be used to determine taxons. Now let me show you the plants we have seen in our adventures. Here is our first site, Ilford site, just north of Sydney, right here. The plants in this site were listed in Jobson's paper as Utricularia dichotoma without the assignment for the subspecies. We don't believe this population was actually included in the genetic analysis, so we are here to identify the species. Using the description of the subspecies and the dichotomous key provided in the 2020 paper, we think this was subspecies maritima. To me, all subspecies look alike, but especially between subspecies dichotoma and maritima are the hardest to distinguish. The key feature we think this is maritima is the shape of the bracts and bracteoles. Maritima has round-shaped spurs at the bottom, but the subspecies dichotoma has square bottom. An Australian botanist, Bose, has done some extensive examinations of dichotoma complex. According to him, bract shapes can also be variable, but subspecies dichotoma in particular, the spurs are reliably square-shaped. I believe he means this. By the way, Bose is the owner of the website fierceflora.com. This site has lots of beautiful pictures of carnivorous plants from numerous locations with detailed descriptions. If you haven't seen it, I highly recommend you visit this site. Since these plants had round spurs, we concluded these are maritima, but only the DNA analysis can tell for sure. Also, please note that we were only able to look at several plants per population, so these features might not represent the whole population. The second site was Casuria Nature Reserve. It was a shallow pond on the coastal plains. This was a known site for Utricularia oppositiflora. Oppositiflora is considered an independent species. The plants here were found only on two spots. One small population was found in the wet peaty substrate by the pond. The other population was also very small and was growing in thick mats of periphytons, fully submerged underwater except inflorescence. were pretty confident that these populations were of oppositive flora. One of the key features was the long, linear lanceolate leaves. Leaf shapes in Utricularia can vary a lot, especially depending on water conditions. Two populations we found both had linear lanceolate leaves, despite one population grew fully submerged in water and the other population grew by the pond in wet peaty substrate. They also exhibited dark vertical nerves on the upper lip, the typical feature of oppositive flora. The third site was in Utricularia National Park in Clearance Valley. We were moving on to the subtropical area of New South Wales, right here. This was a shallow heath swamp over the coastal floodplains. Some parts were covered with beautiful white sands like this. It was an amazing sight. We also saw a huge population of Utricularia uliginosa. The plants here was listed as Utricularia dichotoma without a specific subspecies in iNaturalist. So we were here to ID this population. We concluded that this might be the species oppositiflora population because of the purple veins on upper corolla lip and the slender leaf. The fourth site we visited was called Red Rock. This site was located in a shallow swampy heathy land over the coastal floodplains in the subtropical New South Wales. The population we found here we thought was again either subspecies dichotoma or maritima. In this case, we thought it was not maritima because the bottom of the bracts were rather square than rounded. What bothers me, however, was that there was no apparent spur at the bottom of the bracts and the lower lip was flared, just like Utricularia pollinae. The fifth site was located within the Serpentine Nature Reserve in the heathy swamp on peaty substrate. This was one of the 4 known sites for subspecies Oxyliensis. We found many flowerscapes like these. However, none of them were in bloom, unfortunately. There was supposed to be a subspecies Aquilionia population in sandy creeks nearby, but we were unable to find them. The sixth site, Thomas Lagoon, was in another heathy swamps in the northern tablelands. This was one of the known sites listed in Jobson's paper for the subspecies Novae Angliae. The gibbous base of bracts, linear lanceolate leaves, spur shorter than the lower corolla lip, and the depressed ovovate lower corolla lip all matched the description of Novae Angliae pretty well. This is one of the several Fontana sites within Torrington State Forest, the 7th site. This particular site was near a roadside ditch right beside the main road. Utricularia dichotoma complex can often be seen in roadside ditches, especially in Tasmania. This site was amazing. We saw huge populations of subspecies Fontana and kangaroos. This subspecies was relatively easier to recognize with its unique long spur compared to other dichotoma complex. Also, the bracts and bracteoles were gibbous at the base. We found some unusual flower forms in Torrington State Park. The one on the left was a double-flowered form. I occasionally see twin flowers in Utricularia in cultivation, and I was thinking it was rather an artificial phenomenon. So it was nice to know this could happen in the wild too. The one on the right was a lighter colored foam. This is Evans Head, the last site in New South Wales. This site information was derived from Rob Gibson's previous study. It is a shallow swamp in the northern tableland. Utricularia biloba was also found here. Plants we saw here, we think it might be the subspecies dichotoma. Subspecies dichotoma and maritima, as I mentioned before, resemble each other quite a bit. The ranges also overlap, so it is very hard to distinguish between them. We thought this population was the subspecies dichotoma since the base of the bracts were rather quadrate than rounded. But the bracts were not really gibbous or spurred at the base, so it was rather unclear. At this point, I said farewell to my dear friend Dr. Rob Gibson and flew to the state of Tasmania. In terms of Utricularia dichotoma complex, we visited 2 locations within Lake Pedder area. These locations were from the herbarium records. The upper picture is the Lake Pedder. This is one of my favorite sites in the world. The beaches were covered with colorful jasper quartz, and the Drosera binata grew here were blood red, beautifully contrasting the white quartz background. In the lower picture is one of the habitats where Utricularia bakeri grew. Utricularia bakeri is a species and can be identified by the distinctive purple speckles over lighter lavender color of the upper corolla lips. Also, bakeri is the only species from the dichotoma complex where they have bracts and bracteoles attached in the middle of the peduncles and pedicels. However, I was not able to confirm it because they were too small for my camera for a definitive examination. This is the subspecies Monanthus. This population grows in the roadside ditch right across from the Lake Pedder, where a shallow stream of water were constantly running through it. Subspecies Monanthus most commonly occupy boggy meadows and creeks in alpine regions. Subspecies Monanthus is like a miniature dichotoma. The flower stalks are distinctively short and blooms just above the shallow water level, if not fully submerged. In conclusion, identification solely by morphological differences was extremely difficult. It was because intraspecific variations seem equal to or greater than interspecific variation, meaning the variation between individuals could be bigger than the variation between the species. Also, ecological conditions affect plant morphologies, especially leaf shapes. For example, when fully submerged, the short ovate leaves of some taxa tend to grow linear or lanceolate. Here is the list of the references used in this presentation. I thank my friend and colleague, Dr. Rob Gibson, for his kindness and effort to drive me 800-kilometer round trip to visit amazing carnivorous plant sites. I also thank my friend Lisa Robbie for driving me all around Tasmania, botanizing together. I thank both for kindly sharing his experience, knowledge, and his beautiful pictures. Thank you all for your attention, and I will see you next time.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.